Animals take in water that may be saltier or less salty, may have more ions or fewer ions than their body fluids. They have to get rid of wastes while also managing ion concentrations within levels that are not toxic. If we think about a freshwater crab with eye stalks and mandibles, and we look at cells and urine, hemolymph, and fresh water. Fresh water is low in sodium, chloride, calcium, potassium. Fresh water is also low in wastes, NH3, NH4 plus ammonium ion, usually not very acidic, usually not very basic, and low in any other metabolic waste. The problem that the crab has is that it takes in this fresh water with low ions, and it needs to excrete urine, but it cannot concentrate urine. The urine and the water need to be isoosmotic. In other words, have the same total solute concentration. How does it conserve these molecules of sodium, chloride, calcium, and potassium that are in low concentration in water and are in much higher concentration in the extracellular fluid or in the cells? So if we looked at those concentrations, we would have high potassium, chloride, we'd have low calcium and sodium inside the cell, but high calcium and high sodium outside the cell. Ammonia is constantly being generated, crosses membranes easily. Ammonium, if there's acid present, will form from ammonia, and ammonium does not easily cross membrane, so that's trapped in the urine once formed. What a crab does is produce urine in which it actively moves in waste products. It can adjust how much H plus or HCO3 minus it moves in. Transporters can move in an HCO3 minus in exchange for retention of a chloride, a proton in exchange for retention of a sodium. What the crab does is adjust the urine to be high in NH3, NH4 plus. Depending upon how many other ions it has, it may be high in protons or bicarbonate and other wastes through various exchangers, putting the wastes in and recovering valuable molecules such as sodium, chloride, calcium, and potassium. The waste that's produced is isoosmotic, it's the same concentration as the fresh water that the crab takes in, but it differs in that it's high in waste products. It's low in sodium, chloride, calcium, and potassium. Depending upon what it needs to add or remove from the urine, depending upon the other solutes it has to put in the urine, it may be relatively higher or low in one of H plus or HCO3 minus. If it doesn't need to add more solutes via CO2 that's produced, could be going to the H plus and HCO3 minus via a pathway we've talked about before, or it could diffuse out across the gills as carbon dioxide. The carbon dioxide the crab is producing can be used as a solute in urine, or it can be just released depending on whether the crab needs the solute. Urine plasma or urine hemolymph ratios can also tell us something about the state of an animal. A well-fed animal with a high protein diet will have abundant amino acids that are being converted for other uses and generate NH3 as a waste, and NH3 and NH4 will be comparatively higher in the urine, creating a high urine to plasma ratio or urine to hemolymph ratio of NH3 and NH4 plus. Whereas an individual that has a low protein diet may have lower NH3 and NH4 unless it is being forced to break down its own protein, in which case, even though its diet is low, it's breaking down cellular protein. An animal with a high potassium diet may have higher potassium inside the urine. So an animal on a high potassium diet, which would be plant-based, as opposed to a very meat-based diet, a plant-based diet would have a high urine to plasma or hemolymph K plus ratio, more K plus being excreted. An animal-based or meat-based diet might have a low urine to plasma potassium ratio.